we're now ready to go through the process of meiosis. Very similarly to mitosis, meiosis is also preceded by the replication of chromosomes. So again, if a cell is thinking about dividing, the first thing it needs to do, regardless of whether it's going through mitosis or meiosis, is to replicate its DNA. Now, where meiosis is a little bit different from mitosis is that it actually takes place as two consecutive cell divisions, and we divide these up into meiosis I and meiosis II. So let's maybe go back to our little drawing, right? We had our meiosis. Remember I said that I was oversimplifying the little drawing here. What I drew so far really is just meiosis I, and we have to go through another cell division where we take those haploid daughter cells from the first cell division and we have them, or we divide them, let's say, one more time, and this process here is meiosis II. All right, so we end up with really 4 haploid cells at the end of meiosis in comparison to the single cell division we go through mitosis to create genetically identical daughter cells and only 2 of them. So let's review, right? The two cell divisions in meiosis create 4 daughter cells instead of 2. And each daughter cell has only half as many chromosomes as the original parent, so they cannot possibly be genetically identical to the original parent. We're also going to see the ways in which the daughter cells are actually genetically different from each other as well. All right, so just like what we did for mitosis, we took a really simple cell through this process of cell division. We're going to do the same thing for meiosis. So in this case, we've got another very simple cell. This particular cell is going to have a genome of just 2 chromosomes. So this is a diploid cell. You can see that it's got 2 sets of chromosomes, a set of blue and a set of red, but the sets are really simple, right? There's only 1 chromosome per set. And so this is a cell in interphase. The cell says, okay, I want to go through meiosis. The first thing it needs to do, it needs to duplicate its chromosomes. So it does that during S phase. So here you see now you still have 2 chromosomes. They just happen to be duplicated, right? Each duplicated chromosome is made out of those 2 sister chromatids that are held together very tightly. So the cell's now ready, once it's duplicated its DNA, to go through the first stage of meiosis, meiosis I. In this first stage of meiosis, this is where we have the number of chromosomes. So we take the 2 sets of chromosomes and we separate them. We give one set to one daughter cell and another set to the other daughter cell. So if we go back to our little figure, right, we can add a little disclaimer here that in meiosis I, we are going to separate homologous chromosomes. All right. Now, if you look at those 2 daughter cells, they're haploid. Each one only has 1 set of chromosomes. However, the chromosomes themselves are still duplicated. This is too much genetic information. So this is why we need to go through meiosis II. And meiosis II, it's literally identical to mitosis. This is where the sister chromatids are pulled apart and the chromosomes become unduplicated. So one sister chromatid will go to one daughter cell, and the other sister chromatid will go to the second daughter cell. And this happens for both of the daughter cells that were created after meiosis I. And so in meiosis II, we can make a little note that we are actually separating sister chromatids instead. And that's a really important distinction between meiosis I and meiosis II. Okay, now we're going to kind of quickly go through the different stages of meiosis. This is because a lot of the events that happen in meiosis are identical to what happens in mitosis, right? You have the building of the spindle apparatus, you have the chromosomes condensing, you have the chromosomes lining up at the metaphase plate and then being separated in anaphase. And then in telophase, remember, you do the opposite of what was done in prophase and prometaphase. So all of that is the same. Meiosis I and meiosis II go through those same stages: prophase, metaphase, anaphase, telophase. To make things a little bit more simple, right? Remember, we're going to squish prophase and prometaphase just simply into prophase in both meiosis I and meiosis II. So as we go through, let's say, meiosis and the different stages, I'm not going to focus a lot on the similarities or the things that we already know happen in mitosis. We're just going to stress the differences in meiosis. How is meiosis actually different from mitosis? All right, so in meiosis I, here's where we're going to separate the homologous chromosomes. So the DNA has already been replicated. The cell in prophase is going to do a couple of very unique things as it initiates this process of division. So first of all, in prophase I, yes, the chromosomes condense. Yes, the mitotic spindle, or the spindle apparatus in this case, is being built. But something unique that doesn't happen in mitosis is this process of crossing over. So early in prophase I, you have the duplicated chromosomes pair up with their homologs. They pair up together. This process is called synapsis, and they create what is called a tetrad, right? It's called a tetrad because you've got two sister chromatids from this homolog, two sister chromatids from this homolog, and they're going to come together to form a structure that literally has 4 copies of the DNA, right? So that's a tetrad. Once that synapsis happens, so you can see it down here, you've got the 2 blue sister chromatids of one duplicated chromosome and the 2 red sister chromatids of the other duplicated chromosome. And the sister chromatids are held together by those little rubber band cohesins. And once the homologs pair up, then the DNA is broken. So you can see right here, there are DNA breaks that were made. Now, the DNA breaks are made on one of the blue sister chromatids and one of the red sister chromatids. Typically, a DNA break is bad for the cell, and the cell wants to repair it so that it doesn't lose parts of the DNA, mutations don't occur. Now, the way that the DNA breaks here are repaired is a little bit unique. All right, so first of all, in order to make this repair, the cell needs to make sure that the sister chromatids and the homologs don't essentially come apart. So we already have the cohesins holding the sister chromatids together. Now we need something to hold the homologs together. This is where a structure called the synaptonemal complex comes into play. You can see all these sort of little green structures here. These are going to form this zipper-like structure that's going to hold the homologs really tightly together, kind of like what you see here, right? So there's a zipper structure, there's a synaptonemal complex in green that's holding the 2 homologs together. And this allows in the cell to repair those DNA breaks that were made. The thing though is that the 2 ends that were cut are not the same 2 ends that are stitched back together. So blue doesn't go with blue anymore. In this case, what the cell is going to do is it's going to take the blue end of one chromatid on one homolog, and it's going to attach it to the respective end of one of the chromatids on the other homolog. And so you're going to connect blue chromatid to red chromatid, and red chromatid here to blue chromatid. You essentially are crossing over the 2 non-sister chromatids. And so you're going to mix some of mom's DNA and some of dad's original DNA onto the same chromosome. Now, wherever you have this crossover event taking place, it forms an X-shaped structure. Those X-shaped structures are called chiasmata or chiasma. So anytime you see that on a chromosome, you know that a crossover event has taken place in that part of the chromosome. All right, so that's in prophase. What happens in metaphase I? Well, in metaphase I, just like in mitosis, the chromosomes are going to line up. The difference here, though, is because the homologs are paired tightly together, they're held together by the synaptonemal complex, they've crossed over, so the cohesins are technically holding everything together as well. The homologous chromosomes are going to pair up together at that metaphase plate. So you can see that here, metaphase I, all of the pairs of homologs are lining up together. This does not happen in mitosis, right? In mitosis, each duplicated chromosome lines up by itself at the metaphase plate. Not in metaphase I, right? The homologs are going to pair. If the homologs pair at metaphase I, then when you get to anaphase I, you're separating not sister chromatids, but you're separating the homologs here. This is exactly the whole point of meiosis I, to split the sets of chromosomes. So one set goes to one pole of the cell, the other set goes to the other pole. And that leads us then to telophase I, and then our first cytokinesis. And so here you already see that where the original cell started out with 6 chromosomes, now each of these daughter cells are down to 3. We've already reached the haploid stage for the cells here in meiosis. All right, now if you look at those chromosomes in these 2 haploid cells, you'll notice that they're still duplicated chromosomes. So no chromosome replication takes place between meiosis I and meiosis II because the chromosomes are still duplicated. And so we can go into meiosis II now with the whole idea that all we need to do now is unduplicate the chromosomes. And the nice thing for us is that if you know exactly what happens in mitosis, you've already learned meiosis II. It's exactly the same process. Nothing unique happens here. So meiosis II, all right, you start with prophase II. This is where the chromosomes condense. You build your spindle apparatus. Nuclear envelope is going to disintegrate if it ever even reformed between meiosis I and meiosis II. And you're going to start to have the microtubules attaching to the duplicated chromosomes. Metaphase II, here's where you line up individual duplicated chromosomes by themselves at the metaphase plate. Anaphase II, this is where you separate the sister chromatids, right? This is the whole point of meiosis II, to unduplicate the chromosomes, so to separate the sister chromatids. And then that's going to lead us to telophase II and our second cytokinesis event. And so we end up with now 4 haploid daughter cells at the end of meiosis II. They are also not just haploid cells, but they're definitely not genetically identical to the original parent, which was diploid. But if you take a really close look at the different kinds of chromosomes they've got in there, they are not genetically identical to each other either. So this creates now gametes that are genetically different from each other from that one meiosis event.